Thanks for coming.
Yep.
Appreciate the time, and I hope you'll learn something about microscopy and some image processing and how you can get better images. That's the goal of our talk today. I'm Mark Clymer.
I did that loud and hard to hear you.
Okay, how about if I stand here? Um, I'm Mark Clymer. I'm the Director of Marketing at Datacolor, particularly in its scientific uh, division. And Datacolor's been around for a while, almost 50 years, doing color management mostly for industrial type applications, measuring color and communicating color in things such as textiles, paints, pigments, finishes. A couple years ago, they put their expertise into color management for microscope images. They felt that there was a clear need for this, and this is where how the product that I'll be demonstrating later as it evolved. Joining me today is Dr. Sally Davis from Kansas State. Sally's a 2014 graduate of the NIH National Institutes of Health Comparative Biomedical Scientist Training Program in partnership with NC State University. At the NIH, Sally conducted her PhD research on improving experimental models for the study of human influenza pathogenesis in the laboratory of Dr. Jeffrey Taubenberger. Sally received her DVM and residency certificate in veterinary anatomic pathology in 2007 and 2009, respectively, from NC State College of Veterinary Medicine. Prior to her career in veterinary medicine, she spent 10 years in computer and business consulting, culminating in senior management positions where she was responsible for new business generation, technical leadership of complex systems, integration projects, and development of global curricula in information technology topics. She has a BA in computer science modified with education and a graduate certificate in secondary school science education from Dartmouth College. Sally is presently assistant professor of experimental pathology in the College of Veterinary Medicine at Kansas State, where she conducts research focused on visualizing host pathogen interactions for infectious agents. In addition to her own research, she provides pathology support to the Center of Excellence for Emerging Zoonotic Animal Diseases. Her work leverages both traditional pathology and advanced microscopy techniques, including confocal, multi-label immunofluorescence, and digital microscopy. Sally has a strong interest in the intersection of medicine and technology. Wow. All I can say is I'm delighted to have the pleasure of sharing the podium today with Sally during today's product workshop. So, what I wanted to do first, before we hear from Sally, is I want to kind of refresh your memory with regard to the importance of the microscope in scientific images, and then we'll see some real world experience from Sally, and then I'll provide a live demonstration of a new tool that will help you bring greater consistency and deliver better images that you may acquire with your microscope. And we'll have a Q&A at the end. So if you would, please kind of hold your questions until we get to the end. So the first question is really, do you take the time to set up your microscope every time you sit down? And how long do you think it takes? So, do you think it takes 10 minutes to set up your microscope? Many people think more like 20 minutes. In reality, for bright field imaging, it really only takes about 2 minutes to set up your microscope properly with a little bit of practice. And I'll review some of these practices in the coming slides. The first thing I just wanted to mention is the different optical paths that are in a microscope, and this it relates to color illumination and alignment of the microscope for imaging. So of course we've got the specimen, which is what we look at, but we also sharing the same components. We have the light source. And color illumination actually aligns both of these paths on one another to optimize contrast and resolution in our system. So if we take a look at the anatomy of the microscope, or maybe not that microscope, but this one. Um, Most microscopes that we use have very similar components, and inverted is essentially upside down of this. Um, but then we take a look now at the optical paths, and they do overlap nicely. Key elements are field diaphragm is here at the bottom. Specimen plane is clearly here where the specimen is located on the stage. And then you have other image planes, one up on the sensor, one in the eyepiece, the back of the eyepiece. And then the illumination, they don't, um, even though they do share the same optical path, they don't share the same planes. So an image of your light source is not going to appear in the same place where you might see an image um, of your specimen. But we'll look at both of these as we go through the steps for color alignment. This is, depending on who you talk to, there can be 7 steps, there could be 9, 10, 11 steps. They just break them down a little bit differently. But I'm going to go through each one. Focusing on the specimen is the first, and I think it's kind of obvious, but many people don't focus on the specimen. They go right to the microscope and doing their alignment. But so I'm going to say that focusing on the specimen is taken for granted, and that's already upgrade that out. We're making that assumption. The next is we're going to align the optical path for the specimen. And really what we're doing is we're using the specimen as a reference point. We're in focus there, and we're lining really the light to the specimen, so we're able to flood that specimen with the appropriate amount of light. So the steps are closing and centering your field diaphragm. That's where that may be 2 steps. And there's images down here that you can see. So you close it down, and then you want to center that. You'll also then focus the diaphragm using the condenser focusing knobs. So many times people inadvertently grab the specimen focusing knobs, the stage knobs, and don't see a change, or they go to focus on the specimen and they change their color. So you close the center field diaphragm, focus the deep field diaphragm until the edges of those leaves of the field diaphragm are sharp, and then open that field diaphragm so it's outside the field of view. So that's really simple. You can do this very, very quickly, and it really doesn't change too much if someone's not adjusting the condenser. The next set of steps is kind of the volume of light, if you will, that's hitting the specimen. And the easiest way to do this is to look down into the eye tubes. Take the eyepiece out, look down into the eye tube, and then there's a knob or a lever in front of the condenser to adjust the condenser diaphragm or condenser aperture. And you'll adjust that until it's about 75 to 90% open. This really depends on the microscope that you have and your preference. And then you would replace the eyepiece and proceed with your observation or your imaging. There is kind of a sweet spot for that, and it's the correct balance of contrast and resolution. If it closes down too much, you may seem like you have a lot of contrast, but you really lose your resolution. And I'll illustrate that in a moment. If it's open too much, you're just flooding it with light, and you're not. You've got plenty of resolution, believe it or not, but it's almost too bright and looks washed out. So I'll demonstrate that. Now let's right here actually. So here on the left, you'll see a washed out image, and this is when the condenser aperture is too much open. Far right, you see it's too far closed. Even though it looks nice and contrasty, it's really the improper setting. And I'll call out some features in another slide. In the middle is really kind of the best slide to use, the best setting. So here on the left, you see that the condenser aperture is really too small, and this object that I'm calling out there looks like it has a halo around it. So what you're doing is you're creating a lot of diffraction when you're closing. It gives you the impression that you've got a lot of detail, but you're actually creating it. It's an optical artifact. And it doesn't do you any good, and it's really going to be counterproductive to your analysis. So here's a proper setting of the condenser aperture, where it really is just a speck that's there. What it happens to be doesn't really matter, but just want to identify that that's just a spec proper setting. So what's really important, and most people don't spend the time to do this, is every time you change magnification, every time you change the objective, you need to change those last 3 steps. So you have to go back and remove the eyepiece, adjust the condenser aperture, and replace the eyepiece each time you do it. Now, you can also take note, and some people use a Sharpie and mark their condenser so that they know each time they change objective, they know where the slider or the little wheel or the lever is supposed to be. So that's a really fast way of doing it. Now, it doesn't take that much time. If you do that, you know what the position is. It's 2 seconds maximum. Very, very quick. And the impact it can have on your images is absolutely tremendous. So I'm going to move on to some image acquisition now with cameras, and some of the best practices that I'm not sure that they're always communicated and captured and communicated, but I just wanted to impress that using the exposure adjustment within your camera software is the best way to adjust the brightness of the image. And I like to use the histogram to be my guide for that, leaving a little bit of headspace so that you don't saturate. Anytime you saturate, you're losing data. And I don't know anybody who's willing to throw out data. So, but use exposure adjustment instead of the volume, the light volume, your intensity setting on the microscope or on a power supply. It's a good habit if you do this, whether you have a halogen light source or an LED light source. This way you don't have to think, okay, well, what's my light source? So every time you can sit down with a microscope, could be a different one that you're using exposure adjust. Flat field correction, if your software has it, that's a shading correction. It can happen based on optics, misalignment, it can be some minor vignetting because of couplers to the camera, but you don't always have it, or even the illumination. I have a really bad LED light source that I cannot get centered. It won't center, and it always has hot spots. I always have some kind of shading correction that I have to apply. And even if your software doesn't have flat field correction, take an image of the background. So take your slide off and take an image of that to focus on your specimen. Take your slide off, take an image right there of that background. And then you have that background image that you can use later. So, but if you don't have it, then you don't have it to use. I like to use a white balance for an entire image. Again, I like to white balance without the specimen in place. This way I can tell whether or not I've got contribution of any kind of color in my mounting medium and anything else I'm putting on the stage. I also don't like to use automatic mode because if you've ever watched, you can see that the background changes all the time. And if you're going to try to compare those images, it's just going to become a nightmare because there's a lot more work you're going to have to do in the end. And linear images is important. It's difficult to kind of comprehend, but we want to make sure that our stepping stones, if we would put a grayscale there, are equal steps. We're not taking bigger and bigger and bigger steps, which is common in many imaging systems. And these are the profiles that are put on by many manufacturers to give us a pretty picture, but in reality it can be kind of damaging because camera to camera, system to system, that profile will vary. It will be different, and becomes a real nuisance in the end when you're trying to do some analysis. So really quickly reviewing the acquisition process, you'll do your color alignment, compose your image and focus. You should have already kind of focused, but as you move things around, right, you have to refocus. Do a white balance, as I mentioned, adjust your exposure, and then snap. Any future images, you've already white balanced, so you can skip that process and just continue to adjust as you need. If you change objectives, make sure you change your color. It's so easy, as I said, 2 seconds to do that. It really makes a huge difference. So this is kind of getting into the nitty gritty now of we've acquired our images now and paid a lot of attention to color alignment and the camera settings. But what happens is we go back and we see this, or I took some liberties and adjusted my microscope differently without changing the specimen. But you can see the impact that you can have on the images. And so now you're thinking, okay, what am I going to do? So post processing is very common and it's accepted, in the scientific community. But does anybody post process their images before they? I think almost everybody will. Are you using Photoshop? Image J. Okay. Some people go to. I've seen people buy Metamorph just to go and process their images. I'm like, wow, okay, deep pockets. But even PowerPoint. People will use PowerPoint, go and adjust the saturation and stuff in PowerPoint. But where's the scientific rigor in a lot of this? Well, in a few moments, I'm going to show you a new tool, somewhat new, 2 years, that's really going to get you the results, the majority of what you want to achieve in Photoshop. Will get you there a lot faster and a lot easier. So here's some original images. These were not all taken during the same session because you'll see a variety of some shading and some other artifacts in there. But here's a bunch of original images. One of the tools that we're offering is called Easy Image Prep. And what Easy Image Prep does is automatic white balancing and brightness adjustment in a batch mode. You feed it TIFF and JPEG images, and it'll go ahead and process it with a single click. Most people know about Datacolor ChromaCals from our calibration. Calibration needs a calibration slide, and I'll show you that process as well. But Easy Image Prep doesn't need that. So think about all those archived images, and all you really want to do is quick tuning with white balance and brightness, and this will do it. So I mentioned color calibration with ChromaCal. Even though you may do white balancing, doesn't guarantee you that the colors are correct. Only color calibration will do that, and it's because of the calibration slide, which functions as a standard, color standard in the system. So I'm going to pass the podium now over to Sally Davis for her portion of the presentation. Clicker if you like it.
Mm-hmm. Good morning, everyone. My name is Sally Davis, and thank you very much for the detailed introduction, Mark. And I recently arrived at Kansas State University back in December of 2014, and I inherited equipment, which is very common when you first go to an institution if you're a junior faculty. So the equipment I inherited is up here as follows. It was an Olympus setup. It's actually a pretty high-end light microscope with the X46. It had an older style camera, DP25. They've moved on a couple generations since then. And it had supposedly had the CellSense standard version 1.2. Problem was that the hardware had been cannibalized. A number of the communication boxes off the back were missing. They've been added back here. And this microscope was unable to communicate with the computer. The computer was actually missing, and the CellSense software was absent. So I had to put all that together. And in the process of putting that together, I started looking around for other things I needed, such as monitors, and started thinking about what sort of images I would get off the back of this new system I had inherited. So one of the things that Mark didn't mention is that Datacolor also has a monitor calibration device and software that goes with that. And in speaking with a friend at the NIH, who's also a pathologist, an MD pathologist, he helped me select some monitors. I had the privilege of buying a number of new monitors. I went with a set of ASUS monitors, and I purchased the monitor calibration set initially from Datacolor and used that to make sure all of my monitors looked the same. Because as you probably know, when you look at one monitor, you look at the image on one monitor, you look at it on another monitor, they can vary greatly. So I wanted to get rid of that problem. And in the process of speaking with, this was Stephen Hewitt at the NIH, he also mentioned the color calibration system from Datacolor. So I started speaking with Mark about that, and I got that in-house to try. And I was duly impressed, actually, by that. I'm very excited to use it on my system. This is something that Mark's already mentioned. This is the color calibration slide picture. We usually take a picture of it at 200x, and then you magnify it in their software. And it's very quick once you get used to it. You magnify the image, and you take their matrix, and you align it over the top. And what this allows you to do is to allow the software to see if you've got linear image on all those different colors. And you can see here that initially on my computer, and setup at least, I had very poor linearity. So I messed around with this a bit, and to be honest, was at a bit of a complete loss, and so I contacted Mark to see what was up with my system. Because of course the Olympus guy had just visited and told me everything was perfect, back to default factory conditions, and everything should work fine. So we went through a number of attempts to bring my system into linearity. This is a one-time thing we had to do up front, and we found that instead of a gamma of 1 and a contrast of 1, we had to shift it over to a 0.8 on the gamma and a 0.5 on the contrast to bring the system into linearity. And since that day, I've left it in that state, and it's caused me no trouble ever since. But it is something to note. You need to look at your setup when you first go to improve your images and see what you have, whether it's linear already. And if not, there are adjustments you can make within the vendor software to bring it into linearity, and then you can use the histograms like this to check what's going on. So I actually published recently my first image panel using the ChromaCal software, and I'll talk a little bit later about my workflow with that. I used to use Adobe Photoshop for all of my adjustments, and so I shifted away from that, and I now exclusively use the ChromaCal calibration slide and calibration software for my image post-processing from brightfield microscopy. As mentioned earlier, I'm an infectious disease researcher. I do work as a pathologist in that field, visualizing host and pathogen interactions. This is a recent publication in Virology, which has the full panels, of which I'm going to show you a portion. So when I first captured images for these panels, on the left we have four pictures that are H&E, hematoxylin and eosin, and on the right we have immunohistochemistry for the viral antigen. I mocked up this panel specifically for today. This is an uncalibrated set of images off my microscope. So you can see there are a number of issues with them. They're very dark. The contrast is very poor. It's very hard to see, particularly in the H&E, the viral antigen distribution in these liver tissues. So if we look first at the H&Es, I brought the before calibration slides over here on the left, and then this is using the calibration slide, quickly snapping the matrix in place, which now takes me about 30 seconds, and then batch processing. You can batch process 100 different slides if you've image acquired them through their software. And you can see that even though these were different H&E lots, in fact from 6 months apart in a lab that wasn't actually doing a very good job of calibrating their H&E staining, that they come up looking very similar. And the other thing I like about these images is now you can see the inflammation very clearly. You can see that this is not hematoxylin labeling protein. It's actually blood that's in the tissue there that's not present in the image above. And you wouldn't know looking at them now that they come from very different lots of H&E. I played around with this further, pulling slides from years ago and slides from the present, and you can actually get a lot in that. You can't get rid of an absolute problem if they, as our lab one day, forget to put the hematoxylin on or the hematoxylin stain runs out. You can't make hematoxylin come back. But you can certainly enhance and make the color very true on the images such that they come into much better alignment. If we look at IHC, the key thing with IHC is knowing whether you have a signal or not. I don't have the control slides up here, but this was a very clean immunohistochemistry with very little brown on the control slides. So what I care about here is where's the brown, what cell is it in, and how is it distributed around this tissue. So before calibration, you can see I'm looking at a fairly dim image. Things look a little smudged, and the clarity of the brown is very hard to see. You can't see where it starts and finishes. And over here, I have the IHC where I can actually see it very well. And once again, these are two different IHC runs. These are IHC runs by hand. So although a technician is following a protocol, there's going to be variations on a given day, environmental variations. What is 5 minutes for one person might be 5.5 minutes for another in terms of the chromogen exposure. So I am very happy with the consistent images we get, and we're able to pool IHC runs together in such a way that the panel looks like everything was done in one batch on one day. And it also helps me from the point of view of analysis as a pathologist to be able to interpret the slides appropriately in terms of level of signal. So once again, this is the original panel I started out with before I went through the process of calibration, and then this is the panel that went into the publication. And unfortunately, you're not seeing the true beauty of this because our projection here is suboptimal, but it really makes a huge difference on the slides. So this is a more recent panel. It's been submitted to JID. I do research in pneumocystis, which is a fungus that affects immunocompromised patients, HIV AIDS patients, and it actually is often the cause of death with a lot of these individuals. It causes a very bad pneumonia that over time erodes in its ability to respond to antifungals. So I work with another researcher at the NIH on this, and we were looking at the change in distribution in the number of macrophages in this tissue over time in different treatments. And so the upper row here is untreated but pneumocystis infected tissue, and you can see there are an awful lot of macrophages there, the brown labeling. And then when you treat with caspofungin, which is an antifungal for 21 days, it decreases. And then finally, down here we have the controls. We have uninfected tissues where you see the normal alveolar macrophages that are resident in the lung, and a reagent control here. And this is a serum-based reagent control with no primary. And it gives you a sense of how clean this labeling is. Again, this entire slide was calibrated with the software, and this is the panel that's been submitted recently. This gives you a sense of how I got there, and also the difference between an output from easy image prep and the ChromaCal calibration, which requires the actual use of the calibration slide. So over here is my original image off of a scope that is color illuminated, and other things have been done. And I'm probably not as good about that as I should be, but I do try my best. And over here, we have the easy image prep slide at the top, and you can see just with that step, which is a software improvement, you get quite a bit out of it. But you get even more in terms of the crispness of the color and the edges and the immunohistochemical staining when you do the full color calibration. You see a much wider range in terms of the colors on that lower slide. I did this last night, and this is where I might have Mark jump in for a moment. So basically, I took each of these images into Adobe Photoshop. And if you go into Adobe Photoshop, and where I used to play to adjust my slides, you go to Adjust Levels, and you can play around with the histogram and the range on that. And change the input level such that you get a different output and a different fit to that histogram. So out of the box off my scope, I get this as a histogram. And you can see the image cut does a pretty decent job of this. You have a cut off at the top here, and then when you get to ChromaCal, it's very crisp, and everything is exactly within the full range of the histogram. I don't know if you want to say something else about this right now, Mark. You understand this piece of it much better.
Just one point.
Yeah.
With ChromaCal calibration, we recognize, it becomes apparent that many of the stains and dyes that are used really have a much fuller color gamut than can be displayed, or than we actually see. So it becomes apparent sometimes after calibration, like the DAB in the IHC there is a little bit reddish. That's actually some of this extended gamut that ChromaCal is revealing. It's there in the data. It's just kind of obstructed with the way cameras typically process the images.
Okay, much better than I could say it, definitely. Okay, so one other thing that I find easy image prep just recently very useful for, and I've only started using this tool for about a month or so, is if you notice these images at the bottom, these are actually, once again, just bright field images. This one's taken through a fairly simple microscope, not the one I used for the images at the top. But the other one is simply just a digital camera shot of a plate. So the interesting twist to this is I work in high containment. So I don't have my ChromaCal calibration slide in high containment with me because I'd have to leave it in high containment, which wouldn't be very useful. I'd have to drag, slide in and out, massive disinfection, decon, not worth the while. So while I standardly use the full ChromaCal calibration at my desk and in my laboratory over in BSL2, within BSL3, I snap images with a very basic bright field microscope, ship them out electronically, and then I use the easy image prep to clean this up. And so I had an incredibly dark image where I couldn't see the details of that cytopathic change within those vero cells. And when I used the easy prep, image prep, it brought it right up for me. And then just to complement that, I took this image and improved it a bit too. So it can take any sort of images, and I'm going to start playing with it on some of my gross pathology images as well and see what it can do in terms of enhancing the colors and the ability to interpret those. So I want to go over the workflow I had initially before I started using this software, and then talk about what's changed in terms of time and effort. So as I said before, I was an Adobe person, so I used to do a lot of image capture, and because I wanted things to be consistent, I would spend many, many hours and do one session just to make sure I got consistent lighting, settings, everything across all of my microscopy images. And if I didn't do that, I would have a bit of a nightmare later on bringing everything into alignment. Then I would go through an image adjustment within Adobe Photoshop, and if I was lucky, I would, by eye and looking at the adjust levels, get something that was acceptable. But often then when I go to a second or third image, I'd have to back up and readjust. I'd spend a lot of time going back and forth, trying to get acceptable compromised levels, manually adjusting them, and then I would run those across. And I never got particularly good at batch within the Adobe software. I know it exists, but I would tend to just do these things by hand. So very, very time consuming in terms of prepping, and a lot of tracking, particularly if I was playing with the adjustment levels of which one I actually was going to go forward with for the image panel. And then I happen to use Adobe InDesign. I find it to be very useful to pull in the finished images and then do all the annotations in there, and that's a personal preference. And I have my stock annotations I keep, like little arrows and bars that I know match the different magnifications on my scope. So I've streamlined my process there already. So moving to the use of the data color products, I made some changes. One of the things I do is I no longer feel bound to a single session when I'm prepping for a publication. I can do multiple sessions, and the only additional thing I do when I take 3 images or 40 images is I take one image at 200x of the color calibration slide. And if I've been fooling around with my scope or changed something else in my environment, I will also do a quick check that it still aligns with the matrix and is linear, so that's an extra minute or so in the ChromaCal software. Then what I do is when I get to preparing for my publication is I pull in that same calibration slide for a given session. I quickly align it, make sure it's linear, and then I click on the batch processing, which hopefully Mark will show you. And what that allows me to do is open up a viewer where I can see all of the images I took in a session. I keep them all in the same folder for my own sanity. And if there's multiple stains, I can look through those in terms of whether I need to adjust saturation or contrast based on a particular stain quality. And then once I have a set of adjustments, sometimes there's 0 adjustments, sometimes their software output is exactly what I want, I apply that to a given stain. So for all of my H&Es in the folder, or for all of my immunohistochemistries with DAB and Hematoxylin in that folder. And that's literally pressing the button, and I'm usually down the hall having a cup of coffee or on the phone or doing something entirely different. And their stuff, I haven't timed it, but several images will go through in a minute, so we can process very quickly, at least on the PC I have. And then what happens is I have all of these organized. So I'll have the original image folders, and I'll have a subfolder that says color calibrated, and I'll have that across all the different session folders, and then I have those to cherry pick from for my image panel at the end. It's a very efficient process. I would say it sped my time up by about 8 to 10 times what it used to be in terms of panel construction. Mark was a little worried because now we got 4 boxes on this flow, but really it's 4 very quick boxes as opposed to 3 very slow ones. So I just want to summarize what I've told you here today in terms of my experiences. I have a serious reduced turnaround in terms of my images, and I get these very consistent high quality images out of the process. I'm able to integrate the images from multiple sources, bar a few difficulties with actual stains being done poorly at the lab level or mishaps at the technical side of the biosample management. And I can use them from different sources, different scopes. I played around with a little bit. We'll be playing around with more, definitely different time periods. And I often put together image panels that are across different tissues, different species, and I still feel there's good consistency there. Simplified workflow process, which I just went through for you. And then I'm beginning to expand my use to these other images I have. So I'm a pathologist. I have gross images. I'm also a virologist, so I have images of plates and Western blots and a whole bunch of other different things. So being able to integrate that and have a consistent composite panel like the one I showed you with the plaque assay and the CPE and in the dish as well as the immunohistochemistry is very valuable to me. And I didn't get asked to put this up here. I really firmly believe that Mark and his other colleague, who also happens to be called Mark, are very good on the customer support side. I don't think more than 24 hours have ever passed when I've emailed or sent them a message asking for some assistance, particularly in the beginning when I was trying to get my scope settings in position. And they're also very receptive to ideas about the usability. Mark mentioned earlier I had 10 years in computer consulting before I went into veterinary medicine. So I have some training in usability and usage of different software. And as I've tried to do my workflow in their software, which may not be how they think of doing workflow, I've noticed certain things that there could be ways they could arrange it or alternate routes through their software. And they've actually been very prompt in later version releases in taking some of those ideas into account and making adjustments. So I'm very happy here to be speaking for them, and nobody's paying me to be here. So if we'll move on to Mark now. And, yep. Yep, you're welcome.
All right, so now I'm going to just take a couple of minutes. I know we're getting a little short on time, so thankfully ChromaCal is very quick and easy to use, so I'll show you just how quickly that does work. I'll start with a demonstration of easy image prep, and then I'll follow with a demonstration of full color calibration. So to do that, let me step out of this, pop open. So this is what the application window looks like when you first open it. This is actually black. Let me start from square one. In fact, let's go back to this. This is what it looks like when you actually open it. It's always looking up for latest version, and so this is what the screen looks like. So first step, I'm going to do easy image prep first. So that's up here in the top menu bar, white balance and flat field correction. So easy image prep does do flat field correction if you have a background image, if you've acquired it. It's a lot easier than ImageJ unless you've already built your macros. But I'll show you that. I'll demonstrate it. So here on my desktop, original images, easy image prep. So here I've got, you don't see them because I'm just loading all the images that are in there. I'm just choosing a folder to use. So here you can see the images that are in that folder, and I can click each one. And without doing anything else other than selecting my source folder, it will automatically do that white balancing and brightness. So you can see this one shifted from this and this. This is a really lousy LED. And this camera does a great job of capturing a really lousy LED. So very cold, very blue, and here on the right, it's been processed. I also have my background image. So you can see this lousy LED and all that shading, the hotspot. Destination folder by default is the same folder. This is something that we didn't mention yet. We make a copy of all the images. We never touch the original. So you can always go back and do something again to the original. So many times in Photoshop, you've probably done this yourself. If you don't save a history or save a copy of it, you've overwritten the original and you're toast. So I'm going to save back into the same folder. Now I do have a background image, as I showed you, and that's right here. So I'm going to choose that background image. And I've got the option whether or not to apply the image for flat field correction or not. So if we're just looking at this background image here in the before preview, and here in the right side, you see it after easy image prep with flat field correction. I can toggle that off, and you can see that it's just been white balanced. Now I'm going to leave it on. I'm not going to bother doing easy image prep for that, but let's look at some of these other images here. There's the before and the after. If I turn off the flat field correction, you can see the impact that has. So I mean, it's a really nice feature, very quick way of correcting your images, white balance, brightness, and flat field. We can also save images, TIFF images, down to JPEG. So if you're concerned about storage, size, then we can do that. What I'll do is I'll just click process images and take a look at another one maybe, and we'll process the image. One other thing before I process, this icon goes to full screen, so you can click that, and in full screen mode, then you see the The processed image, lower right-hand side, shows that it's after easy image prep. Use a space bar to toggle back to the unprocessed image. Escape gets me out of that. So I'll process those images. And processing power really depends, or time depends on the number of pixels. Since this does a pixel by pixel correction, and also the processor, and if you have a solid state hard drive, it's even faster. So ChromaCal is able to process images up to 2 gigabytes. I don't know anybody capturing those unless you're doing whole slide imaging, and then it's 2 gigabytes to 40 gigabytes or more. So we're done 6 images in 6 to 10 seconds, somewhere in there. Now, just to show you where those are, or what they look like, I'm going to go into our viewer first. And I'm going to add the images. I've saved them back into my original easy image prep. So here's all the images that were in there. Here's an original, and here's the processed image. And you'll see in the processed image, there's information down here at the bottom that we didn't see in the original image. Okay, this is information that ChromaCal is saving in the metadata with that processed image. So there's full traceability in this. Anybody ask you what was done? Yeah, it was processed. Look, okay, it really was. We also have some cropping tools here that you're able to use. So you can zoom in, and then if you like, you can move that around then if you want to pan around and kind of position it right. If you want, then you can save the cropped, just that region of interest. So it's something just to make it a little bit easier to do that. This is some of what Sally said. We're very responsive to suggestions from researchers, so we want to make your lives easier. Okay, so that's easy image prep. Now I'm going to show you image calibration using a calibration slide. So I do have another folder here. And here's the image of my calibration slide. When I take it, I like to save the serial number of the slide in the image file. This way I know I have maybe 10 different calibration slides. Because I'll leave them in somebody's lab to use. But I like to, this way I know which one. And the reason why that's important is each slide is serialized. They've been measured during manufacturing to know exactly what the color composition is of each of the patches on the slide. So that makes that also a standard. And these are interference type filters, so they're not subject to typical fading that you would find with plastic film that people have played with. So I'm going to open that up, and this is, depending on magnification you used, it'll fit or not fit in the window. First step is to put the grid in the boxes, which is kind of difficult, but I'm just going to, instead I'm going to zoom. We've got all these flip tools, go back and forth, but white patch goes here where the white corner is. It's easy to remember. I'm just going to click and zoom across like this. Boom, done. So those boxes went from orange to blue. Tells me I'm aligned. You see here, it tells my matrix alignment is good. All right. My linearity is also good. I'll click on linearity. So unlike Sally's linearity, it had a nice curve to it. This is nice and straight. And we're looking at that regression of the red, green, and blue color components of this rightmost column. So it's not a perfect gray neutral density ladder. It's pretty close, but it's difficult to do that with interference filters. So we had to give a little bit of color. But we're looking for a regression of as close to 1 as we can get, but 0.997 is our cutoff. And then also exposure. We'll make sure that this slide in particular is not overexposed. The whole background is black. So if you use auto exposure, the tendency is to swamp it. So when you're first acquiring this image, you just want to make sure that you take a look at this white patch and it's not 255, it's not saturated. I always shoot for 220 to 240. Okay, so at this point now I could do a single image calibration, or I can batch calibrate, and that's what Sally says is this is really the value that what people like. Similar window, right? Looks like easy image prep. So you select your source folder, which is that. Boom, it pulled in my images. that are here. Again, TIFFs or JPEGs. Now it's already doing the calibration, even though I haven't told it where to put the image. So haven't saved it yet. I'll go ahead and choose that same folder again. ChromaCal doesn't overwrite, so we can store it in the same folder. That kind of gives you a quick reference. But here you can see the impact of the calibration. So with calibration, ChromaCal is also doing a white balance and brightness adjustment. So it's very, very consistent. You do have some options here for your own liking. If you feel that it's too saturated, you can turn saturation down. That would apply to this image only, or you can apply this setting out to the entire list. So there's some very quick to return each click saturation. So very quick to I only have a few images here, but if you had 100, 200, you could do that here. So right, so you can turn them all off, you can turn them all on, or you could uncheck or check the ones that you want. Right, so here I'll just do 3. I'll save them down to JPEGs, and I'll go ahead and process the images. So this is color calibration. It's amazing that as fast as it took me to align that grid to the image of the color matrix, the algorithm, the ChromaCal algorithm generated a color profile now that was specific to that imaging session. And that's what ChromaCal is applying to the images. So we processed 3 images. And now let's go take a look. So if they don't appear here, they appear, go into the viewer. They're in calibration. So here's the images, uncalibrated. We call this Elephant Man. Very creative that way. And here is the calibrated view. And here you can see the metadata that is now saved with the calibrated image. So there's a lot more information down to the serial number of the slide, the version of the software, white balance values, saturation values, alignment. We tag everything with sRGB. So we do the calibrations in device independent color space. But then we want to make it appropriate so you can visualize it. So we tag it with sRGB because that's the most common color gamut that's in our displays and our monitors. Okay. So you can also make some adjustments here if you desire. Okay, so that's calibration. Very, very quick. Anybody using whole slide imaging, it's very difficult to calibrate these massive images, but ChromaCal can also be used as a kind of a quality control device. So image the matrix, such as this, and then you can capture the actual RGB values of each of those little blue squares, those regions of interest, and export those. So that would be in a snapshot in time what that device is producing. Now, could be a month or 6 months later, you do it again, you find that maybe there's been a shift, time to call for preventative maintenance. Maybe a bulb needs to be changed or something. So we do have customers who are using that for whole slide imaging. If they do take a lot of images, they want to calibrate something, then they would need to export in TIFF or JPEG. Again, those are the only formats that we're going to utilize. Okay, so that is easy image prep and full color calibration, and I now open it up to questions. Okay.
So you used examples with TIFFs and JPEGs.
Yeah.
Are those only supported image formats, or could you input like PNG?
Only TIFF and JPEG.
Okay. What?
So are you capturing and the actual you capture as ping? Okay.
Yeah.
But we could save the TIFF down to a JPEG.
Okay. Oh, so you can't save it back
Yeah,
to
yeah.
Okay.
The default would be to save it as the same file format, but we can save down to a JPEG. We use a compression of 8 on that, so you get good compression, about 90% savings in space, but still very good quality retention. Okay. Well, thanks for coming. If you want to have any other questions, I'm down in booth number 838.
Okay.
I've got other images there. I've got a bunch of examples on the monitor, and it can always take you through another demonstration if you so choose. I'll have slides with me. You can see actually what the product looks like, along with monitor calibration, which is something that Sally mentioned that I hadn't brought up. So
Thank you very much.
appreciate your time. Thank you very much.